Innovation is the key to the success of our industry and the health of our planet. It's my pleasure to be able to introduce the next two speakers who play a pivotal role in both of these missions. Dr. Chilton with his work in the genetics of essential fatty acids that will enable people around the globe to correct the omega-3 deficiency, and Dr. Lewis with his depth of understanding of clinical nutrition while serving as a colonel in our U.S. military and taking care of our troops. These two gentlemen, with their drive and passion towards improving the health of others around the globe, is a shining example of the future of the omega-3 industry. I'm proud to call them both my friends, and I'm proud to be able to collaborate in many research initiatives with both of them. Dr. Chilton is widely recognized for this work on nutrigenomics. He is a full professor in the Department of Physiology, Pharmacology, and the director for the Center of Botanical Lipids and Inflammatory Disease Prevention at Wake Forest Health Sciences, Harvard Medical Center, Johns Hopkins University School of Medicine, and the University of Colorado School of Medicine. So please join me in welcoming Dr. Chilton.
Thank you, Carrie. It's always wonderful to be introduced by one of your best friends, and Carrie truly is. I want to thank Adam, I want to thank Harry, I want to thank Ellen for this wonderful opportunity to speak. Do I have a hand thing that allows me to move slides? Okay, thank you. That could be useful. I do want to acknowledge the National Institutes of Health for funding my center for the last 9 years, and I'm so grateful to them. I also want to point out one individual, Rasika Mathias at Johns Hopkins. She's a very, very gifted statistical geneticist that just really has played a pivotal role in these studies. So, we really have been focused on this, and really focused on this from the perspective of what's driving health disparities. Why is the Western diet punishing certain segments of the population, certain racial and ethnic groups, certain ancestry populations much worse than others? And that's really been the driver behind our work. And I think when we started this 5 years ago, we could have never imagined that there could be so much genetic diversity in this pathway. For a long time, people have been sitting there and out in there, and the idea is that not much long-chain PUFAs are made from linoleic acid and alpha-linolenic acid. And that's been the pretty much the assumption of the field, and I'm going to show you some data that's going to challenge that. It depends on your ancestral background to the degree to which you can do this. Now, if we just simply look at data, and we've looked now in serum lipids, we've looked in plasma phospholipids, we just looked in 2,800 people in the MESA cohort, we've looked in human livers, autopsy human livers, and you see this, and you see examples like this. And basically what you see is, if you look at different cohorts, what you're going to see is within any cohort, there's going to be a dramatic difference in the capacity of people or their circulating levels of long-chain PUFAs. And you see it in arachidonic acid, and you see it in DHA. So within a population, you see dramatic differences. But when you look between populations, for example, African or European ancestry populations, you see almost double the amount of arachidonic acid and DHA in African ancestry populations. So what's responsible for this genetic variability? Well, we have to look at the pathway, and I'm a simple country boy from North Carolina, so I think of things in terms of pipes. And so if you look at this pipe and you look at the pipe coming down, we've recognized for the 28 years I've been in this field that the rate-limiting steps of getting from linoleic acid and alpha-linolenic acid down this pathway to produce these long-chain PUFAs that eventually go into phospholipids, that eventually come out as these pro-inflammatory products or anti-inflammatory products. The rate-limiting step is in these desaturase steps. They were called delta 5 and delta 6 desaturase. The genes that encode for them are called FADS1 and FADS2. And I'm going to focus most of my talk on these genes. So before I get there, I want just us to understand, and we all get it, but the modern Western diet punishes certain segments of the population much more than others. And you can see it in all kinds of data, but if you look in non-Hispanic blacks or Hispanics, you're going to see that, again, you see these major health disparities. And it's not only in the incidence of these diseases, but it's also the severity of the diseases themselves. And if one looks at prostate cancer, which has gotten a lot of attention at this meeting, one can see that African Americans are almost twice as likely to get prostate cancer than Caucasians. Now, one can also, if they look within the population, the severity of the disease is much worse. Well, we heard a talk yesterday that's got a lot of press, and I'm not putting down anyone, but blacks have more prostate cancer, and guess what? They make more DHA. Wow. I wonder if we should take that into consideration. It may not be an exposure issue at all. It may be a generation issue. So we have got these mixed results. We've got these mixed clinical trials. We've got all of these studies, and we've got everyone fussing and fighting with each other. And you know, we really don't know what we don't know. And what we've assumed is that everyone responds the same way, everyone produces the same amount of these things, and so deficiencies will be the same across the board, excesses will be the same across the board, so treat all humans the same. Well, we can't do that. And there's many reasons we can't do that, and I'm going to show one slide, my good friend Joe, who's down here to kick my butt if I say something wrong, but I'm going to show a slide from his group that really shows that when we think about these things, we have to think about exposure. And a lot of people talk about exposure, but we also have to think about both the genetic variants and the epigenetic variants that control whether or not these PUFAs get down to long-chain polyunsaturated fatty acids, whether or not they get to eicosanoids, whether or not, so all of these intermediate phenotypes down the pathway play a critical role as to whether or not you're going to see these clinical phenotypes. So as I promised, Joe and their group, just a magnificent study a couple of years ago, and basically what they reminded us of is something we already knew. Back in about 1961, and I won't get into the whole politics of that because that'd take the rest of the talk, but there was a dramatic increase in linoleic acid into our food supply. Now, linoleic acid is the precursor to arachidonic acid. So linoleic acid goes up, and this is soybean oil consumption, goes up to 8% of energy. So basically, we go from 2% of energy to 8% of energy as linoleic acid. Alpha-linolenic acid stays the same at somewhere between 0.5% and 1% of energy. So we've dramatically, in a very short period of time, and perhaps there's been no better example in human history of increasing a major nutrient so quickly. So we changed exposure dramatically. Now, I'm showing these data here, but if we began, and again, we've done it now in probably 5,000 different people looking at 4 different cohorts, but if we look at the capacity to get down these pathways, and if we compare African Americans and European Americans, and this happens to be the 2 of the cohorts, the PREDICT cohort and the DHS cohort, Well, African Americans, if one looks at products and precursors, they get down the pathway much better. Again, as I showed you earlier, they make arachidonate much better. African ancestry populations do it. And if you look particularly where they seem to be doing it better, it's the conversion of DGLA to arachidonic acid. It's the FADS1 step. Now again, if you look at, and we've shown this data a long time, but if you look at PUFAs, you're going to see the same thing. You're going to see this enhanced conversion, this enhanced ability to make DHA, and if one looks at precursor products, the ultimate product, DHA, the precursor ALA, if one looks at intermediate steps, DHA from EPA or DHA from DPA, one sees very, very significant differences in these populations. So again, dramatic changes in exposure and dramatic differences in populations with their capacity to produce long-chain PUFAs. So one of the general assumptions, and it's been a great, and Bill Lands, who is my hero, really played a critical role, but there's been a general assumption, and it's based on 5 trials that have been carried out in largely Northern European populations. And basically those trials say that 2 to 3% of energy can get down this pathway. Well, what that means is, and as Joe and many of people have shown, is, okay, if you increase the amount of linoleic acid to alpha-linolenic acid here, and only 2 to 3% can get down the pathway, guess what? You're going to make a lot more arachidonate, and you're going to make a lot less EPA and DHA. Because only 2 to 3% can get down the pathway, and you're trying to force 10% down the pathway. Now, 10% of energy. Now, I'm going to argue, and respectfully so to my colleagues, that probably in African Americans, and we're doing the trials right now, that's probably 6 to 8% of energy. So that's going to change dramatically with that population. So again, what you have is these functional, so you have exposure, and you see this enhanced capacity to produce these long-chain PUFAs. Now, we've worked with this for a long time, and in particular, we focus on the rate-limiting step. We focus on the FADS genes. Now, the FADS genes, I'm sorry, I didn't mean to do that, but they reside on, they reside on chromosome 11, in the 13q region of chromosome 11, and what you see is FADS1, which was the original delta 5 desaturase, FADS2, which is the delta 6 desaturase. They sit right beside of each other, and they're transcribed in opposite directions. So we started out 6-7 years ago, and we started out with a homogeneous population in Tangiers off the coast of Maryland. The reason we chose the population is it had exactly the same diet. A boat came over every day and it gave them the same diet, and so once they got the same diet, we didn't have to worry about changes in exposure. So basically what we showed was that variance in that FADS cluster, and in the FADS cluster, and in particular this variant which now has been shown to be the most predictive, RS174537, in a recent GWAS, not so recent, but Tanaka and colleagues showed that it has a p-value of 10 to the minus 49 of predicting long-chain PUFA levels, and that was in a GWAS study. That is as stronger association as you ever see. So your ability to get down this pathway is determined by the gene variants that sits in that FADS cluster. Now, we showed in this paper that at RS174537, if you have the GG variant, then you're going to make a lot of arachidonate. You're also going to make a lot of DHA, by the way. If you have the TT variant, you're not going to make very much. And really where the action is right around this FADS1 step, this conversion of dihomogamma linolenic acid to arachidonic acid. So it's a very complicated slide, and I hate when people do this, but basically what I've done is put all the genes, it's a candidate gene study that we're just doing and we're about to publish with MESA, but I put all the candidate genes that could participate in the elongation and desaturation to form long-chain PUFAs, and basically this shows that most of the action is happening in the FADS cluster. So all the stuff that we believed all along it was the rate-limiting step, it really is. So then we began to look at differences between African Americans and European Americans. And showing you data that I just showed you a second ago, African Americans much higher levels of arachidonate than European Americans. And again, it focuses around this AA to DGLA step in the conversion. And again, these are p-values of 10 to the minus 48. So they're dramatic differences, dramatic racial differences in PUFA ability. This slide is very complex, but there's one simple message from it. It doesn't matter if you're African American or European American. What matters is your genotype. And basically, we had to oversample in this particular study in GeneStar at Hopkins. But basically, what we showed was if you're GG, you're going to make a lot of arachidonate, a lot of DGLA. If you're a TT, you're not going to make much. And it doesn't matter if you're African American. What is driving this effect is genotype. So, why do African Americans, if the same genotypes produce the same amounts, why does that matter? Why do African Americans make more? Well, it comes down to a frequency issue. So, in about 5,000 people in Piedmont, North Carolina, if we look at the frequency of the GG variant to the GT and TT, this is the high converted variant, 81% of African Americans have that variant. European Americans, only 47% have the GG variant. So the differences, the reason we're seeing the dramatic differences have to do with the frequency of the variants. So the question now becomes, okay, in African Americans and other populations, how much get down the pathway? Now I know I'm running short on time, so I'm going to rock and roll, so I'm going to warn you. So we've just published this paper about a year ago, and basically what we showed, and it was in agreement with a haplotype that was published by Amor and colleagues, but basically we looked in the 1,000 Genomes Project. We analyzed almost 1,100 people in the 1,000 Genomes Project. We collaborated with Josh Askey and his supercomputer at the University of Washington in Seattle. And basically, to make a long story short without showing you the data, what we showed was that 85,000 years ago, or roughly 85,000 years ago, there was a variant that was derived on the African continent. And it instantly went to fixation. And we saw some of the highest selective evolutionary values that we had ever seen with this variant. Now, if one looks to see the distribution of that, and if one focuses on RS174537, what one will see is that variant, the converter variant, throughout Africa. It hit 85,000 years ago, went to fixation. For reasons we don't clearly understand, and we can talk about later, but out of Africa populations 50,000 years ago began to lose that variant. And again, if you look at allele frequency as you move across Asia, you're losing the variant. Now, interestingly enough, when you get to the Americas, you're 100% TT, you're 100% non-converters. That raises a profound question of what Native Americans populations do when mother's pregnant and they're eating Western diet. No capacity to produce long-chain PUFAs. So, again, I'll go from this to this slide. So what? So we make more long-chain PUFAs. Does that translate to anything? Well, we just completed a study with Bob Murphy doing LC-MS of whole blood eicosanoids. And basically what we showed in this study is if you're the GG genotype, not only do you make more arachidonate, but your capacity to make leukotrienes is much greater as well. So it translates not only to making more of the fatty acid, but it translates to the biological products. So one of the problems I've always had with this data is RS174537 sits out here. Now that's always bothered me because that's out here in a region 2 minutes. Oh my God. It sits out here in a region that really can't impact a whole lot. So one of the hypotheses, or the hypothesis that we came up with, was this was actually tracking an epigenetic site within the FADS cluster. To make a long story short, we looked at almost 160 livers from autopsies from African American and Caucasians. And basically what we did is we did 485,000 epigenetic scans of the entire human genome, and now we've done that with another 1,100 people as well in demo. But basically what we showed was that RS174537 was simply a surrogate that was marking an epigenetic site that sit right in the middle of chromosome 11. We then went on to look to see where that epigenetic site was located, and it was sitting here located in an enhancer region between the two proximal promoters of these two genes. So this, whether or not you could methylate this site, was indicating whether or not those two genes could be transcribed. And RS174537 was marking that epigenetic site. And again, enhancers put the DNA in a three-dimensional structure and allow transcription to take place. And this particular epigenetic methylation site sits right in the middle of that enhancer. So again, this epigenetic site, much more so than even RS174537, determines your capacity to produce those long-chain PUFAs. So as I said, the FADS cluster genotypes are serving as surrogate biomarkers for methylation sites that are sitting in critical regions between the FADS1 and FADS2 genes that essentially give rise to molecular intermediate phenotypes and clinical phenotypes. So the last slide I'll show, Carrie, is that these epigenetic sites are going to be important. Now, there's been a couple of studies that just came out, and we've just done a study in 1,100 people in the MESA study, and in all of the, what we did is monitor 485,000 methylation sites throughout the human genome, and we looked throughout the human genome, and there are 3 sites that sit in elongase 2, the first committed step in omega-3 biosynthesis, that are the most associated with age of any gene in the entire human genome, with p-values in our MESA study of 10 to the minus 139. So these promoters for the EOVL2 step, the first committed step to omega-3 biosynthesis, are being highly impacted by age. And we don't see any other gene that is so impacted by age. So again, the last question is, yes, there's great variability. Are we doing clinical trials on the right populations? We talk about omega-3 deficiencies, but are we really choosing the populations that are deficient? Are we doing trials like we saw yesterday and saying, wow, prostate cancer, elevated omega-3 levels, must be exposure? Probably has nothing to do with exposure. So we don't know what we don't know, but we have to know more to make sense out of this science, especially under the circumstances of where there is such dramatic genetic variability between populations. This is my lab. Again, love Raska Mathias, and she's my cohort in crime, and I wouldn't be anything without her. She completes me, so thank you so much.